So what about the application of DNA technology in forensics? And this has been huge, a huge change in the whole way we do forensic analysis because of the recombinant DNA technology advance. So here's an example of using DNA from a defendant, blood from the defendant's clothes, and blood from the victim, and being able to analyze it in a way that you can see that the victim's blood was actually on the defendant's clothes, that the blood on the clothes here does not match the original defendant's blood. So in order to use this technology, we have to understand a little bit how we got there. So let's talk a little bit about the procedure called polymerase chain reaction. So this process was developed by someone called Kary Mullis in 1983, and he won a Nobel Prize for his work in the late 1990s. In order to do PCR, all you need to know is a few base pairs that surround a gene of interest. And these base pairs are maybe about, you need maybe 20 base pairs. And then what you do is you can have them made for you by a biotechnology company. So all PCR is doing is like making an amplification of one gene of interest within the cell. And so you need a very small piece of DNA, a target sequence to start off, something as small as a swab of a cheek or some hair found at a crime scene. And in order to do this, it just involves a couple of interesting little steps. So you first have to make your DNA single-stranded. Then you're going to add your primers, and the primers are specific to this region of the DNA, so they will bind there. And then you allow this sequence to then be replicated. And what will happen is DNA replicates in the 5' to 3' direction, so you will be replicating your gene of interest. And after a small amount of time, you're going to take this reaction and then heat it up to a higher temperature, so that what will happen is that the DNA will become single-stranded, and then you can repeat the process again and again and again. So you would make it single-stranded, add primers, replicate, and now you'll be replicating more pieces of DNA. And so within a very short amount of time, you can have millions and millions of copies of your sequence of interest. So what I have to show you is actually interesting short show in the by Learn Genetics about the actual process of PCR, or polymerase chain reaction. So this is a short little virtual lab that we will do together, and I will walk you through it as we go. So in a cell, we have about 3 billion chemical base pairs. And what happens is we can amplify a very small region of that genome. So we first need to extract some DNA from the cell's nucleus. What PCR does is allows us to amplify up a small segment into 100 billion identical copies in a matter of hours. Kind of like the fastest photocopy machine in the world, because we could never make that many copies with our photocopy machines if I was going to copy one page of your work. So in order for it to do, we need a sample. So it could be something like a bacteria, it could be a virus, it could be matching criminals to crime scenes. And in order to do it, we just need to have something that has some of that individual's DNA on it. It could be a sample of blood, skin, saliva, even some hair follicles is enough. So what we're going to do is we need to actually take our DNA and put it into a PCR tube, and then we're going to run it in a little thermocycler. So these are special pipettes that take up very small amounts of DNA. So there's my DNA of the sample that I want to amplify. And now I need to add my primers. So the primers are going to be flanking the gene of interest, and I can have these chemically synthesized by a laboratory once I know the sequence I'm interested in. Obviously, I'm going to use a sequence that I know is going to amplify the region of interest and give me specific bands that I can look at. Once I've added my primers, I need to add the next few things that we need to make DNA, and that's going to be my A's and C's and T's and G's. So I have a mixture of my 4 nucleotides that I need to add into my test tube. So when you're making DNA, we need the nucleotides, and then we also need the enzyme, DNA polymerase. Now this DNA polymerase is very special because it was isolated from a bacteria called Thermus aquaticus, and this bacteria can live at really high temperatures. Our DNA polymerase would not work at temperatures above, say, 38 to 40 degrees Celsius. But since these bacteria live at very high temperatures, their enzyme operates at that very high temperature. So now all I need to do is actually to take my tube and put it into my thermocycler. And I need to kick the dog out. Okay, so let's see what happens in the thermocycler. I'm going to press the start button. So first thing I need to do is heat my DNA sequence to 95 degrees, and at 95 degrees DNA double helix gets broken into single strand DNA. Then I lower my temperature to 50 degrees so that my primers will find their matching pieces on the DNA of that individual and line up. Next, I'm going to add my DNA polymerase and heat up the sequence to 72 degrees Celsius. This is the temperature that this DNA polymerase would work at. And all the individual nucleotides would be copied into the right place. And so now I have my first copy. Now I heat it back to 95 degrees, make it single-stranded. My primers will come and bind when I lower the temperature. Heat it back to 72, the temperature of this polymerase, and now I've made another copy. And then I can just keep running this cycle over and over again until I have many, many copies of my fragment. So my initial fragments will continuously be made, but eventually I'll only be making the main fragment that I have. After 30 cycles, I have over 1 billion fragments that contain my target sequence. And then I'm going to use this process called gel electrophoresis. I can run out my fragments, so each of these would represent a different reaction. One would be blood from the tube, blood from the defendant's shirt. This would be victim's blood, and so this would all be in all 1, 2, 3, 4, 5 different polymerase chain reactions where I'm amplifying specific sequence around my target fragment. And then when I compare them by gel electrophoresis allows you to separate out DNA fragments according to size. So maybe I had a mixture of 10 primers, so I amplify these different fragments. And you can see very clearly that the size of the fragments that were on the defendant's shirt match the victim's blood. This technology is so powerful that it has led to the exoneration of many, many innocent victims. Some of these have even been on death row for crimes that they did not commit. And the person who first used this technique in the late 1990s were actually the same lawyers who defended O.J. Simpson. And after that case, they went on to create the Innocent Project. And they've been helping people who've been incarcerated for many years prove their innocence and be released. So in summary, when we look at sciences and the medicine, many Nobel Prizes have been awarded, many of them due to advances in this recombinant DNA technology. The major goal is to improve the health and wellness of the world population through medicine, nutrition, and research. And many of our genetic diseases are actually not due to single genes, but combinations of genes. And so the future is now to be able to predict which patients are predisposed to heart disease, to cancer at an earlier stage, and then help them change and alter their lifestyle so that those diseases do not manifest in them. And you'll see that this is a very exciting time to be studying biotechnology. And just on studying microbiology helps you understand this whole wide open field of biotechnology that is happening today.